Assalamu alaikum. This is Marwan Al-Halabi. I am a biology teacher, and I would like to describe the easiest way by which biology is explained. In my point of view, people did not advance and prosper until their methods of understanding science developed. However, the traditional method of indoctrinating information has proven its failure by producing completely dependent nations. Nowadays, information has become available to everyone in the easiest way, with the click of a button. Therefore, the best way to reach our goal is to encourage the youth generation to search for information independently. This makes them lifelong learners, giving them the skills of creativity and excellence. For, for this purpose, I suggest to apply the guided inquiry-based learning method, during which learners will follow the scientific method to draw out knowledge. So let's take a look on how the learning activity will take place. Your book consists of 4 units, for example. Each part consists of chapters, and each chapter consists of lessons containing documents through which you should draw out your information. Now, first of all, let's consider your ebook, which is the same as your book that you use at school. On page 12, first, you can see the table of contents, which consists of parts which are units. And each unit or part has a title, for example, reproduction and genetics, and another unit whose name is immunology. Immunology, for example, is the unit 2 or part 2 of this book, and it consists of 3 different chapters. Each chapter consists of lessons, and each lesson is called a group of documents. So let's consider page on page 142, for example. This is page 142. It is a lesson which is called Documents 3 that talks about induction of the specific immune response. But before this idea, I would like to introduce each unit, how it begins. Look, when we when part 2 or unit 2 begins, they state the general title of it. Let's say it's immunology. They put an introductory paragraph or an essay that gives an overview about this unit, and they put the name of the chapters that included in this unit. We begin مثلا, for example, in chapter 6, which talks about the role of and components of the immune system. They put an introduction which we should read carefully, and then they put the large problems that we will discuss, they will discuss in this chapter. For example, how is the biological self determined? How is the immune system organized? And how does the immune system recognize invaders? And to solve these problems, they they put some lessons, and in this chapter we have 7 lessons, and each lesson consists of different documents by which we can, by the guided inquiry method, we can draw out the needed information to be understood by this lesson. For example, lesson 3 or documents 3 uh, begins with an introductory paragraph that contains the problem. Let's say, how does the induction of the specific immune response takes place? So these documents, for example, this lesson consists of document A, B, C, and D, four documents, and each document shows either a certain idea or an experimental result to be interpreted and suitable to draw out the necessary information. And by the end of each lesson, we will see a group of questions which are named probing documents. These questions helps us to draw out information, the suitable information from each of these documents. And the teacher may put is free to put suitable questions to be answered by our discussed with you. Of course, these questions serves the idea of learning the skills of how the scientific method goes on, and teaches us how information can be drawn from experiments and documents. يعني, as a biology student, you should be able to know how scientists work and how they draw information. Let's take this example. For example, document A talks about Moisier experiment, which was done at 1967. This experiment, if we want to describe this experiment, we say that we remove the spleen of a certain mouse and we take the cell suspension from the spleen consisting of macrophages and lymphocytes and distribute them to three different culture media. The first one will contain only macrophages. The second one will contain both macrophages and lymphocytes, and the third one contain lymphocytes alone. And then they introduced sheep red blood cells to these three culture media, and 4 days later the results are obtained where no antibodies are being produced in the first and the third culture media, while the second one only could produce antibodies against sheep red blood cells. So this is a technique by which we describe an experiment. However, if we want to analyze this experiment, we should begin with the results. So, for example, we can say for this experiment, no antibodies are being produced in the first and the third culture media after adding sheep red blood cells for 4 days to a culture medium, culture medium containing macrophages or lymphocytes taken from a suspension cells of a spleen from a mouse. However, antibodies are being produced with the same addition of sheep red blood cells for the same time, but in the presence of more macrophages and lymphocytes together, taken from the same mouse. So by this way, we analyzed this experiment. So what can we draw out? So as a conclusion, we can draw out that antibodies production necessitates the presence of macrophages and lymphocytes. So we concluded that the cooperation of macrophages and lymphocytes is necessary to produce antibodies. So with this experiment with this experimental results, procedure then results, we could learn how to describe an experiment, to analyze an experiment, and to draw out suitable information. But before we end up this video, I would like to introduce the glossary at the end of the book, which contains all the necessary information